presentation. Hi. Okay, thank you very much. So I'm a postdoc at the University of Cambridge, and I'm working on this Cryo-EM project where we have tomographic projections with a lot of noise of proteins, and we want to determine the structure, like the protein structure in each of the images. So our assumption is that we have a heterogeneous protein, which means that there are some flexible parts that can move, and then in each image we have a different conformation of the protein. So we want to determine this structural variability. So we make these assumptions. We assume that the set of possible conformations is a compact manifold, and then we also assume that we have solved the homogeneous cryo-EM problem, which means that we have like the average conformation, and we also know the orientation of its tomographic projection. So under these assumptions, we want to determine the structural variability. So first of all, we represent our proteins as a point cloud, where each point in the point cloud represents the position of one of the C alpha atoms in the backbone of the protein. And then we represent each point cloud by a volumetric function, which is the sum of Gaussians centered at each of the C alpha atoms. So our goal is to construct a map which goes from the manifold of conformations, which I call M, to the set of parameters. In this case, the parameters are the rotation angles between the points in the backbone. So how do we construct this map? We want to construct this map as a linear combination of the eigenfunctions of the Laplace-Beltrami operator on the manifold. So here we have two tasks. One is to compute the eigenfunctions of the Laplace-Beltrami operator on the manifold. The other task is to compute the coefficients of the eigen decomposition. So for the first task, the bad news is that we don't know the manifold. So we cannot compute the eigenfunctions, the Laplace-Beltrami operator. But we have a data set with a lot of images, and we can construct a graph Laplacian, which can be seen as an approximation of the Laplace-Beltrami operator on the manifold. And then we can compute the eigenfunctions, eigenvectors of the graph Laplacian, which we use as an approximation of the eigenfunctions. Then we need to compute the coefficients, and for this we formulate this variational problem in which we have the Yj as the images, like the tomographic projections, and then this long expression is like the parameterization of our protein using the eigen decomposition, and we minimize over the possible coefficients. And by solving this non-complex problem, we obtain an approximation of the coefficients, and then we obtain like the different conformation for each of the images. So we have used this approach in a toy model in 2D. So this is not a protein. This is the two-dimensional structure with two hands and a box which can move. The hands can also move up and down. And we want to determine, so we have a synthetic data set with a lot of these kind of structures. And we want to determine the structure in each of the images. So what we have is the tomographic projections. These are the clean projections. We add noise, and then we apply our approach, and then we obtain this is the original image, the reconstructed image, and also the atomic structure. There are some examples. And now the next step is to try this approach in three-dimensional structure with really proteins. And thanks for the attention.
Our next speaker is Subhadip Mukherjee, and he's going to be talking about deep learning for image reconstruction in CT.